120 million years ago, a creature that had descended from a hunting wasp landed on a flowering shrub in the dry heart of a vanished southern continent and did something no predator does. Instead of seizing prey, it gathered powder, brushing the dust of one bloom onto the next. That single act of pollen gathering, not hunting, not stinging, was the first bee at work, and it would end up reshaping the entire surface of the planet. The land beneath its wings was part of Gondwana, the vast southern supercontinent that held the bones of the world before the continents we know had pulled apart. This was an arid place, a landscape of early flowering plants pushing into a world that had been ruled by ferns and conifers. And in that dry expanse, the first bees were already at work, already binding themselves to the flowers, already beginning the long arc that would lead, more than 100 million years later, to the hum of a wooden hive box in a backyard in Arkansas. We call them bees. We think we know them. They are the most studied insect on Earth, the one we keep, name, breed, and depend upon. The honeybee in particular feels almost domestic, almost familiar. A creature we imagine we understand the way we understand a dog or a horse. We were wrong about almost all of it. We were wrong about where bees came from, wrong about how fast they spread, wrong about how the honeybee in your garden actually got there, and wrong, most unsettlingly, about how much of its ancient diversity we have quietly erased without ever noticing. The genetic history hidden inside the bee is far older and far stranger than the textbook story, and only in the last few years have scientists been able to read it. This is what the DNA actually shows. Let us begin. For most of the 20th century, the deep origin of bees was one of the great unanswered questions in evolutionary biology. Everyone agreed on the broad outline. Bees had evolved from wasps. This much was clear from anatomy and from the fossil record. At some point in the distant past, a lineage of predatory wasps stopped hunting other insects and began collecting nectar and pollen instead. They traded the hunt for the harvest. They became the first pollinators in the modern sense, and from that single dietary revolution descended the more than 20,000 species of bee alive today. But where this happened and when was a matter of guesswork. The fossil record of bees is frustratingly thin. Bees are small and fragile. Their bodies do not fossilize easily, and the ones that do survive often arrive crushed, partial, or trapped in amber in ways that obscure as much as they reveal. For decades, researchers leaned on the geography of living bees to reconstruct the past. The northern hemisphere held an enormous diversity of bee groups, and so it seemed natural to assume that bees had originated somewhere in the north, perhaps across the ancient landmass that would become Europe, Asia, and North America. Each theory had its evidence. The northern fossil sites were richer and better studied. The most familiar bee families seemed concentrated in the north, and the earliest widely accepted bee fossils came from northern deposits, which made a northern-cradle seem like the obvious answer. Some of those celebrated specimens were entombed in amber, like the bees preserved in the deposits of the Baltic and of New Jersey, golden time capsules that seemed to point the compass firmly northward. But there was a methodological flaw buried in this reasoning, and it was the same flaw that has haunted the origin stories of so many animals. Researchers were reading the present to reconstruct the past. They were looking at where bees are diverse today, and assuming that diversity marked the place of origin. They were treating the modern map as a faithful copy of the ancient one. And the fossil record they relied upon was not a neutral sample of the past. It was a sample of where paleontologists had dug, where rock of the right age happened to be exposed, and where fragile insect bodies happened to survive. The north looked like the homeland, partly because the north was where people had been looking. To find the real answer, someone had to stop reasoning from geography alone and start reading the genomes themselves, comparing the genes of living bees across every major family, and anchoring those comparisons against the fossils. That is exactly what one of the most comprehensive studies of bee evolution ever attempted set out to do. The work was led by Silas Bossert, an assistant professor in the Department of Entomology at Washington State University, together with Eduardo Almeida, an associate professor at the University of Sao Paulo in Brazil, working with a global team of researchers. They sequenced and compared genes from more than 200 bee species, drawing on hundreds to thousands of genes at a time to make sure the evolutionary relationships they inferred were real and not artifacts of a single misleading gene. To gather that material, the team worked across continents, collecting specimens from 400 locations and pulling additional samples from museum collections, the kind of preserved drawers of pinned insects that hold decades of fieldwork. Then they did something the older studies could not. They cross-referenced those living genomes against the traits of 185 different bee fossils, including extinct species, building a genealogical model of where bees had lived across deep time. As Bossert's co-author Elizabeth Murray, an assistant professor of entomology at Washington State University, put it, this was the first time researchers had broad genome-scale data spanning all 7 bee families at once. It was, in many respects, the broadest genomic study of bees ever assembled, and it was designed to settle the question of origins by sheer comparative force. The headline finding overturned the northern-cradle assumption completely. The first bees did not arise in the north. They arose in the southern hemisphere, on the ancient supercontinent of Gondwana, in its arid western regions, more than 120 million years ago, during the early Cretaceous period. For the first time, as Bossert said, there was statistical evidence that bees originated on Gondwana. Bees, it turned out, are originally southern hemisphere insects. This single finding rearranged the entire picture, because if bees arose in the south and only later spread north, then the rich northern diversity we see today is not the signature of a homeland at all. It is the signature of an invasion, a later spreading out from a southern origin into the rest of the world. The map had been read upside down. But here is where the story becomes strange. The timing of the bee's origin places it shoulder to shoulder with one of the most important transformations in the history of life on land, the rise of the flowering plants. For most of Earth's history, the dominant plants had been ferns, cycads, and conifers, plants that reproduced without flowers and relied largely on wind to carry their pollen. Then, in the Cretaceous, flowering plants began their explosive ascent, and the early bees were right there in the arid heart of western Gondwana at the very moment this revolution was unfolding. The relationship between bees and flowers was not a slow, gentle drift. The study showed that bees diversified faster and spread wider than anyone had previously suspected. As the flowering plants radiated across the continents, the bees radiated with them, each pushing the other forward. The flowers offered nectar and pollen, and the bees offered transportation for the flowers' genetic material. Neither could have conquered the world alone. Together, they remade it. The forests, the meadows, the orchards, the very fabric of the terrestrial world we live in, were co-authored by this partnership that began on a vanished southern continent. There is a tropical fingerprint on this story, too. The research traced the early bees not just to Gondwana broadly, but to its warm, arid zones. And many of the most ancient surviving bee groups still cling to the southern hemisphere and the tropics today, in places like the dry interiors of South America and the deserts of Africa and Australia. These are not coincidences. They are the embers of the original fire, the living relatives that stayed close to home while their cousins fanned out across a changing world. When the researchers mapped where the oldest branches of the family tree still live, the needle kept swinging back to the south. And here, the story becomes darker, because the deeper you trace the bee lineage, the more you realize how much has been lost along the way. The Gondwanan world where bees began no longer exists. The supercontinent itself broke apart, its fragments drifting to become South America, Africa, Australia, Antarctica, and India. The arid landscapes where the first bees flew were torn into pieces and scattered across the globe. Entire bee lineages rode those continental fragments into isolation, evolving in separate directions, and many of them vanished entirely, leaving only the faint statistical shadows that the genome-scale models can now detect. The 185 fossil species the team examined are merely the survivors of preservation, a tiny fraction of the bees that have come and gone across 120 million years. The DNA cannot tell us exactly what those vanished bees looked like or how they lived. It can only tell us that the family tree of bees is far bushier in the past than the living branches suggest, that the diversity we see today is the remnant of something once far greater, and that the bee, like the dog and the horse before it, carries inside its genome the ghosts of lineages that no longer exist. Now narrow the focus from all bees to the single species most of us actually picture when we hear the word the western honeybee. Because if the deep origin of bees was misread, the recent history of the honeybee turns out to be even more surprising, and it has been hiding in plain sight inside the maternal DNA of ordinary backyard colonies. The honeybee carries a particular kind of genetic evidence that makes it ideal for tracing ancestry: mitochondrial DNA. This is genetic material passed down only through the mother, from queen to daughter, generation after generation, in an unbroken maternal line. Because it is inherited along a single thread, mitochondrial DNA is often used to track lineages in animals and humans alike. And in honeybees, it sorts the species into distinct evolutionary lineages that scientists label with letters. There is the North Mediterranean C lineage, which includes the familiar Italian honeybees. There is the West Mediterranean M lineage, which includes the older European dark honeybees. There is the African A lineage and the Oriental O lineage, and others, each a separate maternal branch that has been evolving in its own corner of the Old World for an immense span of time. Here's the first unsettling fact about the honeybees of the United States. They are not a representative sample of this ancient diversity. They are something far narrower. In a study highlighted in the journal Frontiers, researchers with the United States Department of Agriculture's Agricultural Research Service analyzed the genetic diversity of American honeybees using two mitochondrial myocyte DNA markers, examining 1,063 bees from hobbyist and commercial beekeepers across 45 states, the District of Columbia, and the territories of Guam and Puerto Rico. What they found was alarming. The nation's managed honeybee population rests almost entirely on a single evolutionary lineage. 94% of U.S. honeybees belong to the North Mediterranean C lineage. The entire remainder of the genetic diversity was a sliver, 3% from the West Mediterranean M lineage and 3% from the African A lineage. That was it. An entire continent's worth of managed pollinators, the insects responsible for a vast share of American crop pollination, drawn down to essentially one maternal branch of a species that holds many. Read that again. 94% from one lineage. And it grows starker still. According to the researcher Mohammed Al-Baraki, 77% of U.S. honeybee populations are represented by only two haplotypes, two specific maternal DNA signatures, while hundreds of haplotypes exist in the honeybee's native range in the Old World. Many of those Old World haplotypes have evolved over millions of years in their native lands and have developed astonishing adaptations along the way. The American honeybee, by contrast, is a genetic bottleneck, a population leaning its entire weight on a tiny fraction of the diversity its species spent millions of years accumulating. The consequence is a quiet vulnerability. Low genetic diversity means a population is less equipped to weather new diseases, new parasites, and new environmental stresses, because the genetic variation that might contain the answer to a future threat simply is not present. This is not an abstract worry. American beekeepers already lose a punishing share of their colonies most winters, hammered by the varroa mite, by viral infections, by the murky cluster of failures gathered under the name colony collapse. and a population this genetically uniform has fewer hidden cards to play against the next emergency. As Albaraki warned, those millions of years of adaptive traits should be considered for incorporation into U.S. honeybee stocks before it is too late. The pollinator that underpins a significant portion of the American food supply is genetically far more fragile than its ubiquity suggests. And yet, buried inside that narrow, modern population, researchers found something remarkable: a ghost, a trace of a lineage that was supposed to have been replaced and forgotten. This discovery came not from a grand origins project, but from ordinary disease surveillance. Alan Solansky, a professor and insect geneticist with the Arkansas Agricultural Experiment Station, the research arm of the University of Arkansas System Division of Agriculture, set out with his former graduate student Dylan Cleary to map the pathogens and parasites infecting managed honeybee colonies across Arkansas. They sent collection kits to registered beekeepers and received samples back from 110 beekeepers around the state, representing 540 colonies across 47 counties. The goal was practical: find the diseases. But while testing the mitochondrial DNA to identify the maternal lineages of the bees, they uncovered a hidden genetic history nobody had been looking for. Most of the Arkansas bees were, as expected, the C lineage that includes the Italian honeybees. They found smaller numbers of the A lineage of Africanized bees and the O lineage of Oriental bees. But the surprise was the discovery of the M lineage, the genetic strain of the earlier European dark honeybees scattered among the C lineage samples. These were the dark honeybees that had served as the standard domesticated pollinators in America for roughly 200 years. The bees the original European colonists brought across the Atlantic to a continent that had never known the western honeybee before. The western honeybee is not native to the Americas. Every honeybee in the New World descends from colonist stock, insects carried over on ships, and for 2 centuries the dark European bee was the workhorse of American beekeeping. Native peoples in some regions are said to have called the unfamiliar newcomer the white man's fly, a small phrase that captures just how foreign this now ubiquitous insect once was to the continent. Then it was largely displaced. As beekeepers came to favor the gentler, more productive Italian C lineage bees, the dark honeybee faded from the managed landscape, replaced so thoroughly that it was widely assumed to be effectively gone from the American population. But the maternal DNA in those Arkansas colonies told a different story. The dark honeybee was not entirely gone. Its mitochondrial signature, passed down quietly through unbroken maternal lines from those first colonial bees, was still present, still circulating, a living genetic memory of the founding pollinators of a continent. The DNA cannot tell us precisely how those old maternal lines survived 200 years of replacement and commercial breeding. It can only tell us that they did, that the bee humming over an Arkansas clover field may carry, in the genetic material handed down from queen to queen, the unbroken thread back to the dark honeybees that crossed the ocean with the earliest colonists. There is one more layer to the strangeness of the honeybee, and it lives inside the colony itself, in the very machinery that decides what each bee becomes. A female honeybee larva can develop into a sterile worker or a fertile queen, and which fate she meets is not written in her DNA sequence. Genetically, workers and queens can be identical. The difference is governed by something subtler, set in motion partly by what the larva is fed, the rich royal jelly that tips a developing female toward queenhood. Researchers, including Christina Grosinger, the Publius Virgilius Maro Professor of Entomology and Director of the Huck Institutes of the Life Sciences, have probed how genes inherited from the mother and genes inherited from the father are switched on and off inside the developing bee. In mammals and plants, this kind of parent-specific gene control is usually mediated by chemical tags placed directly on the DNA, a process called methylation. But the honeybee, it turned out, does not play by those rules. The team found that DNA methylation does not perform this function in bees. Instead, the control appears to lie on the proteins around which DNA is wound, the histones that package the genome into a structure called chromatin. Chemical tags on the tails of those proteins seem to determine whether the mother's genes or the father's genes are expressed, and ultimately whether a larva becomes a queen or a worker. The bee builds its castes, its entire social order, through a genetic mechanism distinct from the one our own bodies use. And even now, researchers are still working to understand it fully. What does this mean for the bee drifting across the flowers in your garden right now? It means that the small insect you have always treated as ordinary is one of the oldest and strangest stories on Earth. Bees did not come from the north. They arose in the arid heart of a vanished southern supercontinent more than 120 million years ago, and they conquered the world by co-authoring the rise of flowers themselves. The honeybee you keep, or buy honey from, is not a faithful sample of that ancient diversity, but a narrow remnant of it. 94% descended from a single maternal lineage, leaning its enormous importance on a genetic foundation far thinner than anyone assumed. And inside that narrow population hides a ghost, the maternal line of the dark honeybees, the first colonists carried across the ocean, still passing silently from queen to queen, 2 centuries after the world declared them replaced. The bee at your feet is not a simple creature. It is a survivor of continental breakup, a partner of flowers, and the carrier of lineages we thought were lost. 120 million years ago, on a dry stretch of western Gondwana, something that had once been a wasp landed on a flower and gathered pollen instead of hunting. We do not know which insect. We do not know which flower. We do not know which morning the long partnership truly began. But the descendants of that decision are still here, moving across the blooms outside your window, carrying inside them the genetic memory of a continent that no longer exists and a history we are only beginning to read. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.